going to have hear from Ralph Baric, North Carolina, who's going to tell us about synthetic genomics of SARS.
So I also would like to thank the organizers for inviting me to talk. It's been a wonderful venue and a real pleasure to be here and capture some new ideas that can be tried out on viruses. So the general themes that I'm going to talk about today are a basic short introduction into SARS biology, talk about the synthetic resurrection and reconstruction of a variety of zoonotic SARS viruses and their applications in therapeutics and vaccine design. I'll also touch on codon deoptimization as a way to attenuate SARS pathogenesis, but I certainly haven't gone to the depth of studies that Eckert's group has done. And then I'll talk about rewiring SARS coronavirus transcription circuits as a universal strategy to attenuate viral pathogenesis. So this is a schematic diagram of the SARS coronavirus particle. It's about 100 nanometers in diameter. It contains a helical nucleocapsid inside. It contains a single-stranded plus polarity RNA genome. This nucleocapsid structure is surrounded by a lipid bilayer that contains several viral glycoprotein spikes. The important ones for today's talk include the M glycoprotein and the E protein, which are shown right here. These proteins are absolutely essential for maturation and release and the production of virus particles. So keep that in mind. The second viral protein of interest for today's talk is the S glycoprotein gene, as shown right here, which gives the virus its unique appearance in the electron microscope. It's the viral attachment protein that binds to the receptor to mediate docking and entry into the cells. It regulates tissue tropism, species specificity. It contains a large number of important neutralizing epitopes, and it's the principal component of protective immunity. Now, if you tear this virus particle apart and look at the viral genomic positive stranded RNA, it's about 30,000 base pairs in length. They're the largest plus polarity RNA genomes in nature. The first 20,000 base pairs or so encode the replicase proteins, whose job it is to replicate the genome, and express subgenomic messenger RNA that encode these downstream open reading frames. Now, these downstream morphs encode the structural genes like the S, E, M, and nucleocapsid protein that I just mentioned that were present in the virion, and a variety of accessory orphs. To get these expressed in cells, you find a nested set of subgenomic messenger RNA. They're arranged from the 3' end of the genome so that all the sequences in the smallest message are in the next largest message, and so on and so forth. This organization allows different open reading frames to be placed at the 5' ends of different messenger RNAs so they can be efficiently translated and expressed in cells. Now, about 70 nucleotides shown here as these little blue boxes that is derived from the 5' end of the genome. So these are discontinuous stretches of RNA that are joined during transcription. And these little red box elements right here are absolutely essential for mediating the joining of these two sequences. This will become very important later on in the talk. So keep the concept of TRS elements, the little red box elements, as being essential for regulation of messenger RNA synthesis. Now, I'm going to talk about synthetic genomics in the context of a platform to control emerging infectious diseases. If you think about emerging viruses, they have tremendous potential to cause high morbidity and mortality and severe economic hardship. Good examples are HIV, the 1918 flu, SARS coronavirus, H5N1, chikungunya virus, probably most recently. Now, it's clear that zoonotic introductions are increasing, and a large number of these viruses have large reservoir pools of animal strains like SARS and H5 that are maintained as sort of heterogeneous swarms of pools of viruses, of which some one may actually emerge in the future to cause epidemic disease in humans. This raises a conundrum in terms of how do you protect the public health against a future occurrence that is hard to predict? How do you develop drugs and therapeutics against an unknown from this heterogeneous swarm of variant strains that will actually work against the unknown that will emerge in the future? And how do you target your scarce resources? And so we think platform technologies like synthetic biology, phylogenomics, structure modeling, high throughput sequencing really will provide a platform And our goal is to use SARS coronavirus and its large pool of zoonotic viruses as a model to develop rapid response platforms to protect against this broader heterogeneous pool of variants. We also would like to develop strategies that would attenuate all family members so that you could rapidly develop vaccines for the public, overall public health. Now, a lot of beautiful work in Malik's lab, as well as other labs here in China, worked out the basic molecular epidemiology of the SARS coronavirus. The virus originated most likely in bats. It probably jumped into civets or humans, and in that context, set up a transmission cycle between civets and raccoon dogs in marketplaces and humans who frequented those marketplaces. Over time, it selected for strains that were more efficient at infecting human cells and recognizing the human ACE2 receptor for docking and entry, leading to the 2002-2003 epidemic, which caused about 8,000 cases and 800 deaths. Now, if you do phylogenetic analysis of the isolates that were sequenced, you find they fall into two broad categories. This line indicates the strains underneath that line were identified during the epidemic, and you have early phase isolates, primarily of January 2003, after a variety of super spreading events. You had middle phase isolates shown here by this group of isolates, and then following infected physician who came to Hong Kong and resulted in a super spreading event that transmitted the virus to the rest of the world. These are considered late phase isolates. A vast pool of heterogeneous zoonotic strains, however, reside up here. Most of these were, in fact, none of these were ever actually successfully cultured. They actually exist as sequence signatures in silico. So one of the immediate goals that we became interested in during the outbreak was to develop a platform of viruses that captured the heterogeneity that exists within the family of viruses. And to do that, we identified sort of representative strains. For example, early phase isolate Urbani we had in the lab. Middle phase isolates were CUHKW1, GZO2, was an early phase isolate, animal isolates like from civets, SC16 and HCSC6103, a raccoon dog isolate, which is shown down here, the sporadic 2004 human case, GDO3, and a couple of other viruses. Sequences were identified as good candidates that would capture all the diversity that had been identified within the sequence pool. This shows the sequence, that's the sequence variation within the spike glycoprotein. If it's a blue box amino acid, that's an animal-associated residue. Early phase changes are shown here in yellow, middle phase changes shown in orange, and late phase changes shown in red. In addition, in 2004, there are a variety of other animal strains as sporadic human cases were identified that had additional variations shown here in white. And it's interesting as much of the variation actually falls within residues or regions that neutralizing epitopes were been identified. And in fact, although the number of residues that are changed aren't that great, you can actually reduce neutralization titers by about 20-fold with some of these variants. Now, importantly, Farzan's group showed that the two key changes during the epidemic were this lysine to asparagine change at 479 and a serine threonine change at 487 that drove animal adaptation to human strains. And so keep that in mind. So without access to this pool of strains, we decided to synthesize basically a portfolio of spike glycoprotein genes of about 4 kb each, and then use a molecular clone for the Urbani epidemic strain that we had built in the lab, basically replacing the Urbani spike one at a time with these various spike glycoproteins from different phases of the epidemic. Now, all of these viruses were viable except for the SC16 variant. This actually can't recognize the human ACE receptor, so it didn't grow until we made mouse cells that constitutively express the civet ACE2 receptor. And once we did that, this virus could grow well. Before we had those cells, since Farzan had identified this 479 change as the key residue change, we actually built a recombinant virus with that change, and that virus could be cultured. Interestingly enough, two of these, the GZO2 and the HCSC6103 viruses, actually caused lethal infection in aged animals. They produced an ARDS-like disease. SARS caused ARDS in humans, or predominantly in aged populations. That's acute respiratory distress syndrome. It's a clinically devastating end-stage lung disease with about a 50% mortality rate. So these are actually some of the first real good animal models for SARS infection. Now, these viruses replicate, the human epidemic strains replicate efficiently in human airway epithelial cultures. These are cultures that are derived from cells lining the trachea of transplant patients. You can actually culture them on liquid air interfaces, and they take on the architecture of the human airway. SARS coronavirus, all the epidemic strains actually like to infect ciliated cells, and these epidemic strains replicate very efficiently. The animal strains, however, do not. And this is just some fluorescent microscope images showing the cilia of the ciliated cells with expression of the SARS nucleic acid protein from an epidemic strain on the ciliated cells, and the zoonotic strains, SE16 and the HCSE6103, can't replicate. However, if you passage these viruses on human airway cells in culture, you can actually rapidly select out variants that can replicate efficiently in human airways. Those viruses actually do not contain the mutations that had been predicted by Mike Farzan as being, which were clearly responsible for the 2002-2003 epidemic. Rather, we saw changes at positions 442 and 472 and 479 that mediated the cross-species transmission event. So in reality, we've done this a couple of times. There's actually several pathways by which the zoonotic SARS could actually adapt and recognize the human ACE receptor. What's interesting is that the epidemic strains actually efficiently use both the human ACE receptor and the civet ACE receptor. When we in vitro select for human adapted strains on human airway cultures, they actually lose the ability to recognize the civet receptor. So what this data suggests to us actually is that SARS had existed in a transmission cycle between humans and civets actually probably for several years prior to the 2002-2003 epidemic, allowing the virus to gain the capacity to recognize both receptors. Okay, so those were the easy ones to do. Oh, the application. So now that we had a large panel of variant viruses, we could use those for therapeutic testing or for vaccines. In this case, I'm going to show you an example of about 30 human monoclonal antibodies that were derived by Antonio Lancevecchia. If you take those 30 monoclonals and test their ability to neutralize all the strains that we've made in the laboratory, you find some that only neutralize Urbani, some that neutralize all human strains, some that neutralize some civets, but not all the animal strains. But you do end up with 4 antibodies that neutralize all strains that we have in our portfolio, including the ones that were in vitro adapted on human airway cultures. Importantly, if you select for escape mutants, these antibodies select for changes in different locations of the receptor binding domain. And in fact, 3 of them actually don't overlap. The S109, the 230, and the 227 don't overlap. And so these represent good therapeutic cocktails that would capture most of the diversity that exists in SARS and probably work in potential patients that would be infected during future epidemics. And these also actually protect young and aged animals from lethal infection. That was a paper published by Baric in a couple of papers. Now, the civet and the raccoon dog strains were the easy ones. The true reservoir for SARS is within bat populations. So if you look at a phylogenetic tree, the human strains are shown here in red. The civet strains, raccoon dog strains are shown in purple. But the real variation is within the bat strains. The reservoir, it's thought that these were probably the reservoir for the emergence of the virus. Now, these are about 80 to 90% identical to SARS. They can't be cultured, and they exist as sequence signatures in silico. There's also extensive variation through the replicase and elsewhere in the genome. So when we decided we were going to synthetically resurrect the entire virus. Now, before we started, it's important to note, this is a cryo-EM reconstruction of the SARS glycoprotein spike. Notice that there are 3 receptor binding domains shown here with bubbles that actually engage the ACE2 receptor. If you look at the sequence, the receptor binding domain shown here in yellow, there's a large amount of sequence variation, especially within the contact interface residues that engage the human ACE receptor. And in fact, there's only 4 of 13 contact interface residues that are retained in these bat strains. So we don't actually think it's going to use the human ACE receptor to get into cells. In fact, we think we're going to have a tough time culturing this virus. Now, it's important to note that the SARS-RBD that was identified has been proposed by a couple of groups that it may have been introduced by recombination processes from unknown strains that haven't yet been identified, and that led to the initial cross-species transmission events. Now, to get back to the issue of in silico sequences, they actually represent hypothetical viruses. Most synthetic viruses that have been resurrected to this point, actually we knew that the sequence was infectious. In this case, we actually don't know which of the sequences in GenBank were infectious, if any. So to do that, with an error rate in GenBank ranging from about 1 to 500 to 1 to 10,000, depending on who did the sequence, we had to do extensive bioinformatic analysis to identify what we thought was the likely consensus sequence. None of the strains that we actually saw we thought were completely correct. Some of them had deletions in the sequence at the 5' end, so we had to make some educated guesses. The basic approach that we build coronaviruses using our molecular clone is shown here, with the SARS clone shown in blue. The clone is broken into 6 about 5 kb pieces. Each piece is flanked by bagel 1 restriction endonuclease sites. These are class 2S restriction enzymes that recognize a palindromic sequence, but cut and leave asymmetric ends. This allows, since these ends are asymmetric, they actually will not concatamerize like classic sticky ends left by restriction enzymes, but rather they become directional. So if you end clone A with a bagel site that leaves 1 3-nucleotide overhang and the 5' end of the B fragment with the complementary 3-nucleotide overhang, they go together directly. You change different bagel sites at each piece. And this allows them to assemble up into 30 kb chromosomes like Tinker Toys. Now, the synthetic bat genome that we made using Blue Heron and BioBasic basically is interchangeable with the Urbani clone. The only real difference was that we broke the F fragment into 2 pieces so that we could play with the receptor binding domain easily if this thing didn't turn out to be infectious. And in fact, we made this clone, we built the full-length cDNA, we drove transcripts, electroporated that into cells, and we could see evidence of replication by the synthesis of subgenomic messenger RNA, but we couldn't culture the virus and we couldn't pass it from cell to cell. So clearly there was probably a defect in entry. To solve that problem, we used literature data that has suggested that RBD domains of coronaviruses may be interchangeable between species. So we took the human, the Urbani epidemic receptor binding domain, that's 210 amino acids in length, and dropped that into the bat genome backbone, producing a chimera with the receptor binding domain driven from the epidemic strains. Now, when we built that clone, drove transcripts and electroporated that into cells, we got a virus that could replicate quite efficiently. This is just some growth curve data showing, I think the black box is the Urbani wild type and the white circles or open symbols are the bat viruses. You can see they replicate exactly as good as the Urbani epidemic strain at low and high multiplicities of infection. They recognize the human ACE2 receptor in DBT cells at low and high multiplicity infections and grow just like the epidemic strains. And they also retain the ability to use the civet ACE2 receptor just like the epidemic strains at low and high multiplicity. They also are capable of infecting human airway epithelial cultures and targeting ciliated cells just like the epidemic strain, and they grow to similar titer. Now, if you make antisera just against the bat spike glycoprotein and use that antisera to target the SARS wild type virus, it will not neutralize that virus. So clearly, antisera and vaccines derived against epidemic strains are not going to protect against the bat strains. If you use that same sera against the RBD chimeric virus, you can neutralize it, indicating that there are neutralizing epitopes that exist outside the RBDs. And if you take the human monoclonals that target the epidemic strain RBDs, you can neutralize these viruses quite efficiently. So in summary, certainly synthetic genomics and reverse genetics can be a platform to recover uncultivatable zoonotic precursors. It can be used to synthesize. This is actually the largest synthetic virus to date. It's going to be a short record, but it is a record. And you can use it now to identify broad spectrum antivirals and vaccines. Clearly, the RBDs are interchangeable. This is with an 89% amino acid identity within the spike. It's the minimal domain required to host shift coronaviruses. What is the phylogenetic limits to RBD interchange? We actually don't know. Clearly, the human monoclonals and vaccines targeting the SARS RBD would provide protective immunity against natural isolates that emerge by recombinatorial processes in the future, or unfortunately by deliberate design. So now I want to move to the next part of the talk, which is synthetic deoptimization schemes to attenuate SARS coronavirus pathogenesis. This is the maturation pathway for how SARS gets out of the cell. The nucleocapsids align underneath an intermediate compartment in the Ruffy-R Golgi, where the M glycoprotein and the E protein are expressed. And then the M and the E protein actually drive the assembly of virions, which then mature in the Golgi and then are released from the cell in secretory vesicles, which fuse with the plasma membrane. Now, if you knock out E protein expression, this pathway is still viable, but you reduce virus yields by about 2 logs. And if you knock out the M glycoprotein expression or M and E together, you don't make any virus progeny. So our approach to deoptimize SARS coronavirus, the SARS coronavirus genome, and to attenuate pathogenesis focused primarily on the E and the M glycoprotein genes. It's about 350 amino acids in total. The strategy we used was to progressively increase the number of deoptimized codons. So we started with serine to produce a D-SIR virus, serine-leucine R gene to produce the SLR deoptimized strain, or a 5-set DSLR-VA strain. Basically, the idea is you create a rheostat where you're increasing the number of deoptimized residues and turning down expression of critical proteins needed for release. This is just a cartoon to show the amino acid sequence and the wild type virus sequence here. So like in the 3-set at the serine residue, which was optimal in the case of SARS, we just changed it to the most rare codon. If you look at the statistics here, in general, in the wild type E and M gene, there are about 39 codons that are deoptimized. In the 1-set, this increased to 52. The 3-set, 94, and the 5-set, 134. We also looked at the CPB values that Eckert just talked about. By these values, these are very minimally on the negative side of the codon pair usage. We also made random controls where we scrambled the codon usage, much like Eckert did, retaining the wild type genome organization levels, and we made the 3-set and the 5-set. We made these in a mouse-adapted background so they would be pathogenic in mice. The mouse-adapted growth is shown here in black. The blue lines show the serine deoptimized viruses, which also reach high titers by about 24 hours post-infection. If you look at the 3-set mutants, however, you see about a log and a half reduction as compared to wild type. The 3-set randomized virus and the 5-set randomized virus, in this case, I'm showing 2 different plaques. In the 5-set randomized viruses grew just about like wild type. So just like Eckert had reported, if you randomize the sequence, it really has very little impact on replication, but the optimization does affect final yields. Now, in contrast to what Eckert talked about, we actually deoptimized in the middle of the downstream ORS, which would potentially affect critical sequences that would affect RNA synthesis. We were very careful not to knock out any known sequences, regulatory sequences that make messenger RNA. However, both the serine and the SLR mutant knocked out messenger RNA 6 expression, which you can see right here. These actually were the messages, the genes, the messages that encode the genes that we deoptimized. And so we actually found some evidence of long-range protein, RNA-RNA interactions that are regulatory elements that we're trying to decipher. The 5-set deoptimized strains showed no CPE in culture. If you develop primers to the 5' end of the genome in the leader sequence and a primer down here in the message 6 that encodes ORF6, you can actually identify leader-containing transcripts, which are signatures of the messenger RNAs that are made during infected cells. In these cultures, you can see evidence of message 6, 5, 4, and 3. This is at day 1 post-transfection. Even by day 10, you can still see these transcripts in infected cells, and they continue at that level with about 5 passages at 5-day intervals. But you actually never can actually see a virus. You can never plaque a virus. You can never actually show a virus as they're producing CPE. So 5-set deoptimizations really downregulated the production of infectious virions and the ability of these viruses to produce a significant amount of CPE. We can get around this probably by expressing helper cells that make the M gene, but we haven't done that yet. Now we've looked at the pathogenesis of these isolates in mice. In this case, we're infecting with about 10 LD50. The mouse-adapted strain, wild-type parent strain, kills the animals in about 4 days from severe pneumonia. The serine deoptimized viruses actually are almost as pathogenic. There's a slight delay in loss of body mass, but all the animals eventually die. The SLR randomized viruses are also pathogenic, but again, slightly less pathogenic than the wild-type virus. There are some survivors, but most animals die. The 5-set randomized viruses are, again, slightly less pathogenic. Several animals die, but there are survivors. But the 3-set deoptimized viruses, none of the animals died. There was transient weight loss, and the animals recovered. Virus titers within the mice were actually about the same with the wild type, the single deoptimized codon, or the 3-set deoptimized codon viruses. Okay, so our data certainly is in agreement with Eckerd's work that suggests that the optimization or strategies to downregulate translation of viral genes could function as a universal approach to attenuate pathogenesis. In the case of coronas, we can identify novel transcription regulatory sequences that we didn't know exist. We're trying to decipher that, and these are likely very, very stable. However, in the case of coronaviruses and polioviruses, these are both susceptible to recombination repair. And so we needed strategy to get around that. And so that's actually the last part of my talk, which should take a couple of minutes, in which we devised strategies to alter the transcription circuits of coronaviruses. Now, remember I said in the case of wild-type SARS, these red box elements are absolutely essential to make subgenomic messenger RNAs. If the leader RNA red box element can't communicate with these body red box elements, this virus is dead. So we asked a couple questions. Can we change the regulatory circuits? Can we redesign them? If we redesign them, will it act as a genetic trap that will cause lethal genome incompatibilities following recombination? And what's the impact of the new circuits on pathogenesis? So we made 2 viruses called here CRG and CNG. In the case of the CRG virus, we took this red box element virus and converted it to a blue box element virus by introducing 3 changes at each of these positions. We also made a green box virus that had a completely different TRS regulatory element at each of these locations. Now, these viruses were viable. They grow to 10 to the 7th. They're small plaque mutants, but they grow just as well as wild type, and they produce all the appropriate subgenomic messenger RNAs. And if you're interested, take a look at this paper. So the answer is yes. Does it affect recombination repair? The answer is yes. That's also in that paper. I don't have time to go through the data. The idea is if a recombination event occurs, you end up with viruses with scrambled regulatory elements. So the red box elements can't communicate with the blue box elements, and the virus dies. So it's a genetic trap, and it's triggered after recombination repair. So how does this affect pathogenesis? Now, this is young 10-week-old animals infected with the mouse-adapted virus or wild-type SARS. SARS just replicates in mice. It produces no disease in young animals. The mouse-adapted strain causes a lethal disease within 4 days. We put the CRG in both backgrounds. In the wild-type background, again, we see no weight loss in the animals or death. In the mouse-adapted strain, when you introduce the CRG regulatory, the new regulatory network, you completely attenuate pathogenesis. You look at virus titers, however, virus titers in young animals are pretty much similar to the parent strains. This actually challenges a current sort of an existing paradigm in virology that to attenuate pathogenesis, you have to knock down replication, and that's not the case with these viruses. We also looked in old animals. In the case of old animals, the MA15 virus kills the animals in about 3 days. SARS produces a transient weight loss of about 5% and the animals recover. In this case, the CRG produced no weight loss compared to the SARS parent. In the lethal genetic genome backbone, we saw more extensive weight loss in the older animals. These are 1-year-old animals, but all the animals recovered. Occasionally, we lose an animal. And again, the virus replicated efficiently in these animals. So we think that rewiring the regulatory network of a virus can attenuate pathogenesis. Could it function as a vaccine? So we infected young animals and old animals with either the CRG parental virus, a alpha virus encoding the SARS spike, or with PBS. We vaccinated them one time, waited 28 days, and then challenged them with either a lethal homologous strain or a heterologous strain. You can see that with PBS, all the animals succumb by day 4. In the single vaccinated alpha virus vaccine encoding the SARS spike, you see morbidity, very little mortality, and the animals recover. There is a lot of virus and a lot of pathology in the lungs. The HCSC6103 virus is only lethal and only produces disease in old animals and young animals that just replicated, but not within the CRG. It did replicate some in the VRPS. Now, in the older animals, you can see that the vaccine cocktails via the alpha virus vaccines and PBS, the animals died with the homologous and with the heterologous challenge. And again, the live attenuated rewired viruses protected against both. So in summary, transcription circuit redesign can attenuate coronavirus pathogenesis. We actually think it should function as a universal platform to attenuate current and future emerging coronaviruses of the future. It also protects against recombination repair mechanisms. You can blend this with codon de-optimization to really provide a good strategy to make live attenuated viruses. They protect young and old animals. Since SARS targeted elderly populations, this is currently the only vaccine that works in this population. Field vaccines don't. And I think the combination of synthetic genomics and family-specific universal attenuation schemes can achieve a goal of rapidly producing candidate live virus vaccines within about 2 weeks of identifying the sequence of a newly emerging pathogen. So the people that did this work included Boyd Yount, Barry Rocks, Damon Deming, Eric Donaldson, Tim Sheehan, and Matt Freeman. So thank you.